So this lesson is on DNA packaging and control of transcription. So your key question, how are primary mRNA molecules modified in the nucleus before being translated into proteins, and how are proteins modified after translation? The target objective, overall target objective, students will be able to describe the effect of histone modification on DNA packaging, although we'll be talking a little bit more, but the main focus is to know the function of histones and how they modify DNA packaging. Critical attributes. This is something that you should feel comfortable, you should have accomplished by the end of the lesson. So today I will learn the effects of histone modification on DNA packaging so that I can better understand how DNA exposes itself to be transcribed into mRNA. I know I have it when I can explain the process of acetylation and DNA methylation. So for chromosomes in eukaryotes, DNA is associated with certain proteins to form chromatin. So the proteins in the chromatin are responsible for packaging the chromatin into discrete linear structures called what we know as chromosomes. So the extent of packaging changes during the life cycle of the cell and when the chromosomes become visible during mitosis. Each chromosome includes protein coding regions called genes. So these genes are areas where proteins can actually be made going through that process of protein synthesis. So segments of DNA called promoters and enhancers are found at the start of the eukaryotic genes. These are involved in the beginning processes of transcribing a gene into mRNA. So here, what makes up a chromosome are your two chromatids. So you see there's a chromatid here, and it makes up one chromosome. The chromatin is coiled up, and it does that to reduce the length of DNA overall. Your chromatin consists of, again, the complex of DNA and the histone proteins forming what we call beads on a string structure. So this structure here kind of looks like the nerd's rope candy. Your histone proteins provide a support structure around which the DNA is organized, forming an ordered, compact shape. The nucleosomes include those 8 histone proteins wrapped in DNA, and the nucleosomes is the basic unit of DNA packaging. The nucleosomes have the appearance, again, as the beads on the string. So here, the part of the gene that is transcribed includes areas called introns and exons, and it's enclosed between a start and stop sequence. So this is an indicator of where transcription can actually take place. So here, this is a start sequence. So this is where RNA polymerase will bind to. Your exons are here, your introns are here, and then you have your stop sequence. Well, when we are refining or modifying mRNA, that final mRNA strand, the introns are actually removed. And then we have something that's called exon splicing. So once those introns are removed, those exons will start to come together. So, quick question, what is the role of histones in this process? Is it A, B, or C? If you chose C, you are correct. So the histones is mainly to organize DNA, winding it up so the DNA takes up less space than if it was unwound. So let's talk more about DNA packaging and control of transcription. So how is transcription controlled? Because we definitely don't want mRNA to continue to transcribe that full DNA strand. So of course there's a system in place to make sure that doesn't happen. So regulation of gene expression in eukaryotes is a complex process, beginning before the DNA is even transcribed to RNA. So the packaging of DNA regulates gene expression either by making the chromatin packed together tightly or more loosely. So this affects whether or not RNA polymerase can attach to the DNA and transcribe it into mRNA. So packaging of DNA is affected by histone modification and DNA methylation, which is a part of modifying the modification of histones. So the modifications are considered epigenetic, so above genetics, because they do not involve changes to the DNA sequence itself, However, it helps to regulate gene expression as the cell differentiates and then records its cell's history as it experiences different changes in the environment. So looking at this diagram, histone modification is very important because it helps give that epigenetic control of gene expression. So histones may be modified by either methylation or acetylation of the histone tails. So these are your histone tails here. Back it up. This is a chromatid. This is a chromatid. 2 chromatids equals 1 chromosome. Then you have that coil here of DNA we call chromatin. And these are your histone tails. Okay? So acetylation, you see here, removes the positive charge of the histones, decreasing their interaction with DNA and allowing the chromatin to transform into more of a relaxed structure. So it exposes the DNA. It loosens it up. So RNA polymerase can attach to that loosened DNA, attach to the gene coding, that protein coding area, so on the gene that codes for a specific protein, so that transcription can take place. Now, the methyl group here, that DNA that will attach to the histone tail, the methyl group would bind only to cytosine, so one of the nitrogenous bases that make up the DNA strand. And it can either prevent the actual binding of transcription or cause the chromatin to come together so tightly to where its RNA polymerase can't access it. So this is where this also helps. Even though we have a stop sequence, that methylation and methyl group binding to the cytosine actually helps RNA polymerase from transcribing the DNA further. All right, so describe the effect of histone modification, including DNA methylation on DNA packaging. So make sure you're explaining this in your own words. Okay, so in short, I'm not expecting you to have this on your notes. You can have this in addition to your response, but just having this, you will not get credit for your guided notes. So just in short, histone modification includes loosening and tightening of DNA. Okay, so DNA methylation will tighten the DNA to prevent transcription from happening, and acetylation will loosen it. So changes after transcription and translation. So then again, how a primary mRNA molecule is modified in the nucleus before being translated into proteins, and then how a protein is modified after translation. So there's a modification that takes place after transcription and after translation, and we're going to look at that really quick. So fun fact, human DNA contains about 25,000 genes but produce up to 1 million different proteins. So each gene must therefore produce more than one protein. So this is achieved by both post-transcriptional and post-translational modification. Post-transcriptional modification is when those primary mRNA containing those exons and introns are modified. So we have our introns again are removed completely after transcription, and then those exons are spliced together. There are many alternative ways to splice the exons, and the alternatives create many different variations in the translated protein, and we're going to see that in this diagram here. So you have guanidine nucleotide cap is added to the 5' end of the primary mRNA to protect it from dissolving during transport from the nucleus to the cytoplasm. And you have multiple adenosine nucleotides that are added to the primary mRNA. So these poly A tails, that's what we call them. So poly means many, A means adenosine. So poly A tails aid nuclear export, translation, and stability of the mRNA strand. And this is what you see here. So 3 splicing alternatives create 3 different proteins. Okay, so altering that mRNA strand, that primary mRNA, can be spliced and combined in many different ways, and you see that here. Now, post-translational modification, so this is after proteins have been made, they can be modified by adding a carbohydrate, a lipid, or a phosphate group. So these modifications are involved in completing the protein's functional role. And some proteins are first made as a single polypeptide and then cleaved and reformed into a functional form, such as insulin. So you see that here in this diagram. So how can so many proteins be produced from so few genes? Okay, so the answer is A. Primary mRNA can be spliced and combined in many different ways. Challenge question. So if a human produces 1 million proteins, but human DNA only codes for 25,000 genes, on average, how many proteins are produced per gene? So this one took some math. So each gene will actually produce 40 proteins on average. So we would divide that 1 million by 25,000 to get that 40 protein on average count. So to conclude, histone modification is very important because it helps control gene expression. So we have a process in place that will expose the DNA and a process in place that will disclose, so tighten up and make that DNA inaccessible. Here we have our mRNA splicing. So how primary mRNA can be spliced and combined in many different ways. Knowing that once a protein has been translated, it can be modified by cleavage or through the addition of other molecules such as carbohydrates, lipids, and/or phosphates. So at the end of this lesson, you guys should feel pretty comfortable about about answering the key question below. So in your notes, please answer the question to the key question. How are primary mRNA molecules modified in the nucleus before being translated into proteins, and how are proteins modified after translation?